Sagar sir. Indeed, uh, I'm audible, sir, clearly.
Yes, you are audible, sir.
Hello.
Yes, sir.
Okay. Okay. Thank you. Indeed, it is a privilege to be here as a resource person in this PhD coursework 2025, which is organized by Loknete Venkatrao Hire, Art, Science and Commerce Autonomous College, Panchvati, Nasik. Uh, I sincerely thank respected Principal Dr. B.H. Jagdale, sir, respected Head of the Department and Research Coordinator Dr. T.V. Pawar, sir, Course Coordinator Dr. Satish Aire, sir, IQC Coordinator Dr. Vishnu Adole, sir, ARC Coordinator Dr. Nutan Sadgir, madam, my dear friend Dr. Chobe, sir, and all committee members for giving this wonderful opportunity to interact with a research scholar. In this lecture, my topic is docking tools and application in drug ligand studies. This session will definitely answer the three important questions. What is molecular docking? Why there is need to perform molecular docking? And how molecular docking is performed. Let us start with session. Just a minute. Now the question is this, what is molecular docking? Molecular docking is a computational approach which is used to predict how the small molecule, that small molecules are our synthesized compound and we call them ligands, bind to target macromolecules that are usually proteins or the enzymes. The goal to perform this molecular docking is to find the most favorable orientation of the ligand inside the active cavity of proteins or enzymes, and thereby we can estimate the strength of that binding. We call it as a binding affinity, or also called as it as a docking score. Here is the image. This image represents any protein or receptor or the target, and this is our synthesized compound. We call it as a ligand. This ligand can fit into the active site or the cavity of enzymes, and thereby there is interactions takes place between this ligand and protein. We also call it as a lock and key model because depending on the orientation of ligand, it can fit into the uh, cavity of receptor, that is protein or enzymes. This GIF, that is graphical file, can show this is the ligand and sorry, this is the protein, and this ligand can be placed at the active site of this protein. Wherever it will be have more interactions, at that place it will be stable. Now the question is why molecular docking is needed. It is one of the very important tool for the drug discovery and design, by which we can understand the protein ligand interactions. We can also predict the binding affinities or the docking score. We can analyze enzyme and target analysis, that means the ligand and protein interactions. The most important thing, it reduces the time and experimental cost, and it also supports the structure based drug design. As I said, it reduces the time and also experimental cost, because if we think about the drug discovery and its development, it involves different steps like identify the disease, then isolate the protein from the microorganism that causes that particular type of disease, then find the lead compound against that protein uh, present in that particular microorganism. Then there is a preclinical testing, then formulation, then human clinical trials, and at last there is FDA approval. So if you see these different stages or steps, it requires the time period of near about 17 to 20 years. And along with this, it requires lots of money, or it takes many costs. And what happens, suppose blindly, if you target any compound to synthesize as a drug, what happen? You will take all these uh, stages. You will go through all the stages. It will cost lots of money and also period. And at last, if you find that drug is the synthesized compound or drug is not so potent, then there will we can say that there is a waste of time and money. So uh, this molecular docking is a computational approach. It can use the probable possibility of that drug molecule to show its potency. There is again a complexity of drug discovery. That means if you synthesize any drug, that drug can be called as a good drug whenever it can have this type of properties, like it should be targeted, it should be selective, it should be potent. uh, Targeted means what? It should act against a particular type of enzymes. Then it will be the selective. It is potent. It must be potent means what? With a little concentration, it shows its efficiency. Then it should be physically stable, whatever its physical state, that means form. It should be soluble. Then it should be permeable, because they have to diffuse, or that drug should have a very good permeability. Then it should also metabolically stable. That means once you have taken any drug inside your body, just like food material, if it get digest or metabolized easily, then it will not show its action. Then it should be non-inducing. That means it should not trigger any other type of response in your body. It should be reversible. Reversible means whenever any drug binds with any protein, then after interaction, it should get free from that protein. Otherwise, if it will be there remains, then it may cause toxicity, or it may become poisonous to that body. Then it should be durable. It should also be non-teratogenic. Teratogenic. Again, it should not be teratogenic means those type of drugs that can cause the birth defects. Suppose you have taken any compound as a drug, and it may, if it has a adverse effect on the foetus, uh, so we can call it as a it is teratogenic. Then it should not be mutagenic. That means it is non-mutagenic. It should be patentable, and it should be manufacturable. So these are the number of stages, or we can say that number of criteria that the drug should cover, and then we can say that drug is can be approved by the FDA. So once you pass these stages, and then after covering all these things, sorry, that drug should also have this type of properties. So all this process, that is drug discovery process, is so lengthy, and it should give some positive output. So in order to predict the positive outputs of that particular synthesized compound, we should perform molecular docking, because it gives the probable interactions of your synthesized compounds with the target enzymes. Now, types of docking. There are two types of docking: protein-protein docking, and another one is protein ligand docking. Generally, what we perform in our research, that is what protein ligand docking, where ligand is flexible but receptor protein is rigid. So, according to the cavity of protein, that ligand can take its shape and effectively bind with the active site of protein. There are number of docking programs or softwares available, like Autodoc Vina, Gold, Glide, Doc, FlexAd, then RDoc, SwitchDoc, ZDoc, etc. And after docking, there are some softwares available for the visualization of docking results, like PyMol, UCSF Chimera, then Discovery Studio, DocWiz, Invado, etc. For our study, we are using these two important softwares. Both are academic version. That means they are totally free and easily available on internet for the download. The tools for molecular docking we are using is Autovina Doc, which is fast, efficient, and command line based software. That means it works with the command prompt, and the docking results can be analyzed by using this very wonderful software, that is BioWare Discovery Studio. It has a very user-friendly graphical interface by which we can study the visualization and interaction study of protein and ligand. The workflow that molecular docking involves, that means the steps involved in molecular docking are a protein preparation, ligand preparation, grid box setup, then running autodock, that is docking, and finally analyzing the results of molecular docking that can be done with the Discovery Studio. So, as I said, there are, uh, sir, I'm clearly audible to all of you.
Yes, sir.
Okay, thank you. As I said, the first step is protein preparation. So, uh, there is protein data bank available on the internet on the side of RCSB PDB, from which we can take the desired protein in PDB format. So before that, as I said, we require two important things. First one is the protein, and second one is ligand. Ligand is our synthesized compound. Protein we can isolate, or we can uh, extract, or we can take it from the protein data bank. So after searching the desired protein, we have to prepare that. Before that, we can download that protein from in PDB format from this website. We call it as protein data bank. So from protein data bank, once we will see it in demonstration also, how we can download, or where we will get that PDB file. So this is the protein structure. We can get it from the protein data bank. Then we have to prepare this protein in first of all BioWare Discovery Studio also, and then we have to perform the docking in AutoWinner. Then we have to prepare the ligand. Ligand is our synthesized compound. We can prepare this ligand in a different way, but for our study, we prepare in ChemDraw, then ChemDraw 3D, and we can convert the file format from SDV to PDB format by OpenBabel. That involves some steps we will see in demonstration. Now, grid box setups. While running the Autodoc Vina, you will require a configuration file, and that configuration file will contain, or you have to notify which is the receptor, which is the ligand, which type of file you want as a output file, then the log file, that is result file in text format. Then again, uh, uh, as I said, protein has active sites, and in that active site our ligand get bind. So that active site we have to decide its a dimension of that active site by placing its xyz coordinates. We will see it in configuration file. So after getting protein structure and ligand structure in PDB format, we can take it for the docking in AutoWinner Dock software. This AutoWinner Dock software, after docking, will generate this text file. We call it as a log file. That log file involves the binding affinities of that ligand with a particular or desired protein with its binding affinities value. Remember that these values are obtained in a negative sign. More will be the binding values in negative, more will be the negative binding side. We consider it as strong is the interaction, strongest are the interactions. Suppose for this particular docking, we got two poses, only two poses. The first pose having a binding affinity minus 7.7, and second having what minus 7.0, and the first having RMSD value near about, or not near about, it is exactly 0, 0. So this binding score which having lowest RMSD value can be taken as a binding affinity for that interaction. So I think it's one more one more file. This is a result of another molecular docking. In this case, the ligand when interacts with, while interacting ligand with the protein, you will get a 9 different types of poses with 9 different types of binding affinity. So we will take we will consider only that pose which have the highest binding affinity generally and have the lowest RMSD value. For the illustration, I have taken one example. This is molecular docking I have performed in one of our research paper recently published in a Journal of Molecular Structure. In this case, uh, anti-TB activity was taken against these two compounds, and they both the compounds having nearly same anti-TB value, that is MIC value, and then we have performed the molecular docking for that compound by using the protein structure with the PDB ID. I think it's a 19EX. Yes, it's a 19EX. This one, this one is. I have to check the PDB ID. Okay, this is 19EX. In this case, what happened? These ligands bind with this protein in its active site. There are different types of amino acid residues that interacted with our ligand, and we get the binding affinity value minus 7.7 kilocalories per mole. So these are the interactions. These are amino acid residues. We have to report it in our research paper. This is very impactful technique which increases the impact of our publications. Here is many people when we are go through this learning process for the molecular docking, uh, we uh, uh, we understand many things, just like this protein having heme unit, and this heme unit also act as a cofactor which stabilizes this ligand inside the cavity active site or cavity of this protein. So we have to keep it as it is without removing. We can perform molecular docking. uh, After visualizing its 2D diagram, we can see that this heme unit also have interactions with our ligand. So we can report all these interactions in our result and discussion part of research paper. This is what log file. Here we get 7 docking poses. Generally, we got the 9 poses uh, as I selected, but due to the unavailability of space or due to the presence of this heme group, what happens? This protein cannot take more than 7 poses. So we got here, sorry, 6 poses. We got here 6 poses. Out of this, the first one having highest binding affinity minus 7.7 with RMSD value 00. So we have reported it as a docking score. Another compound interacted with the same protein, and that having carbon hydrogen bond van der Waal interactions. This all type of interaction we got. We can visualize this interaction in BioAir Discovery Studio, which is very good software for the visualization of docking results. Again, these two compounds, 3A and 3B, particularly this one is 3B, shows the interactions with selected protein with a docking score minus 8.2 kilocalories per mole. This is its output file. Here we got only 4 poses. The first one with the maximum binding affinity minus 8.4 with RMSD value 00. We have taken it. After visualizing or analyzing these interactions, we can report it in our research paper in this way. There is 2 compounds, 3A and 3B. We have taken this PDB ID or this protein with the PDB ID 1E9X. Docking scores are minus 7.7 and minus 8.2. These are the key interactions between our ligand and the different amino acid residues of protein with their numbers. For this, there are references. Documentation is available online. Then BioVia Discovery Studio website, where you can get different types of information and relevant scientific literature. Nothing but our research paper. By reading research paper, we can select the desired type of protein with its PDB ID and perform the docking. Uh, this was up to why there is need of docking. Now we will go for how we can perform molecular docking. So far, uh, We saw what is molecular docking. Molecular docking is nothing but it's a computational approach by which we can predict the binding interactions of ligand with a protein. Protein gets fits inside the active side of ligand gets fits inside the active side of protein, and it shows its interaction. Maximum will be the interaction. The stronger will be the affinity of that ligand with particular protein. Now we will go for demonstration how to perform molecular docking. Are you ready? Hello, am I audible?
Yes, sir.
Okay. Uh, so far, if anyone having any difficulty or queries, I will be happy to answer. Yes. Okay. I think there is no any difficulty. Let us see. As I said, the docking requires two important parts. First one is a protein, and second one is ligand. So first we will get a protein from protein data bank. In Google, you search here PDB or RCPS, nothing but protein data bank site. Okay. For this, you have to perform literature survey. Which type of proteins you required? Suppose I am taking here protein 1E9X. So I will put here PDB ID for that protein 1E9X. Okay. Why it is slow? Actually, network is okay. Uh, I have already downloaded this protein. Listen, whatever file is required, you have to keep all that file in C drive. In C drive, you have to create a separate folder named as docking. I have created here a new folder with the name docking. In that folder only, you have to save all that files, because this auto winner docs works with the command prompt or command line software that takes the uh, files pathway. So you have to keep all this in C drive in a particular folder. Here are the different types of PDB IDs or different protein structures. So we required 1E9X. Just click on it. Okay, it is again taking time. Meanwhile, we will see. Okay, I have downloaded this 1E9X. I will take it in download. Suppose you have downloaded. Okay, we got here. Here is two tabs. First one is display file and second one is download file. In download file, you have to download that PDB file, protein structure as a legacy PDB format. Just click on it. The downloading will start. Yes, the protein structures get downloaded in download. folder, we'll see it. What happened? I downloaded that pdb file from download files by clicking on legacy pdb format. I got it. I got it in download. Then I just double click it. It get open in BioWare Discovery Studio. Before that, remember you have to download both the softwares. First one is Autovina Dock and second one is BioWare Discovery Studio. Both software you have to install in your computer or PC, whatever you have. Then this file will get open there only when you have downloaded this BioWare Discovery Studio software. Once you click on that pdb file, this protein file I have downloaded from the protein data bank. It is in pdb format. So just clicking on this, you will get this structure in BioWare Discovery Studio. This is our protein structure. Can you see it clearly? Yes. Can you
Yes,
see
sir.
it? Okay.
Yes, sir.
Okay. Now the first step will start. That is preparation of protein. We will prepare the protein in BioWare Discovery Studio. There is a tab here in the view hierarchy. Just clicking on it, there is one more window will open, or just simply you can click here this arrow. You will get same window. Now remember, these red dots are what? The water molecules, and we while docking we have to remove it. So just click on the water. Whatever you click in this interface, that will get highlighted. So I am clicking on water. All red bubbles, red dots get visualized, and that means they are selected. Just go in edit and delete it, or simply you can press the delete button on keyboard. They will get deleted. Okay. Now this is our protein. This is our protein. This protein having, I can change the background of this file of the interface. Color background, I will take it white. Okay. Now you can clearly visualize. Okay. Here is our co-crystallized ligand. Remember, uh, most of the protein having its inhibitor or co-crystallized ligand. This co-crystallized ligand with this protein, and this is the heme unit. We call it as cofactor, which present in most of the proteins. So what you have to do, you have to place our ligand in the place of this co-crystallized ligand. This co-crystallized ligand is already present in that protein. We have to remove this, and we have to insert our synthesized compound as as a ligand. And after inserting, we have to see the or analyze the interaction of that compound. More will be the interaction, stronger will be the affinity of that ligand with protein. So first of all, you have to prepare the protein. While preparation, you have to remove first water molecules. Then after, you have to select the grid box or the active site of this protein. And for that, just click on the ligand. It get highlighted. Then you have to go here, define and binding site. Define and binding site. And here is current selection. After clicking on current selection, the area gets selected that is covered by the co-crystallized ligand. This co-crystallized ligand is already present in that protein. You have to remove that, and add our synthesized compound. So before adding, you have to select the place. We call it as an active site. For the selection of active site, you have to what? You have to click on define and binding site from the current selection, and just right click on. You will get attributes of SBD structures or the pair. That means you will get the dimension of this active site. Why? This is the first part. We call it as a protein preparation, and that protein preparation involves selection of active site. That active site has a dimension. You have to copy that dimension and paste in configuration file. The three important files you will require for the docking. First one is a ligand file, second one is protein file, and third one is configuration file. Configuration file is just a text file. In that text file you have to specify the x y z coordinates, thereby that coordinates helps the ligand to take its position. So there are three coordinates. First one will be the x, second one will be the y, and third one will be the z. So just copy one by one and paste it in configuration file. That configuration file is also have to keep in a docking folder that is created in C drive. So this is the configuration file I have taken here. Initially there was just was ligand. Then I put here configuration file, which is text file, and this text file having xyz coordinates. So I will copy paste this xyz coordinate. Just select this first coordinate minus 16.20 something. Okay, I have pasted it. Then second minus 2.91, I will paste it here. Already pasted, but for the your convenience, we are doing same thing again. I will paste the coordinate for the z axis also, and then after just go to file tab and save it. Remember, this size of xyz can be taken differently, like some people's taking here 60, 64, whatever. But in our study we kept it as a 20, and here the exhaustive value is 8, which is standard as we go through the literature survey. So we get ready configuration file. We got again xyz coordinates of this ligand. Now what is our next work? We have to remove this ligand. How? Just go in hierarchy tab, and then there is two ligands they are showing. Actually this is not ligand, this is the cofactor, but it is other than this protein structure. That's why they consider it as a ligand. Ligand 1 and ligand 2. Can you see it clearly? Ligand 1 and ligand 2. Hello, can you see
Yes,
it
sir.
clearly? Yes.
Yes,
Okay.
clear.
So 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 what is the active site? Active site is the position of ligand 1. Ligand 1, this one is the co-crystallized ligand. We call it as a co-crystallized ligand. And the second one is, it's a cofactor, nothing but heme unit. So heme unit is required for docking, so we will keep it as it is. And this ligand 1, so- sorry, ligand 2, this ligand 2 is the co-crystallized ligand. We have to remove it before docking. So just click on ligand 2, go in edit tab and delete it. Now I have deleted. There is no ligand. Now there is just a empty space. There is just empty space which is available for the insertion of our synthesized compound. We call it as ligand. Now we have to save this file. Before saving, go to the chemistry. Under the tab hydrogen, add polar hydrogen. We have to add polar hydrogen after removing all the not all the co-crystallized ligand. Add polar hydrogen. Polar hydrogen get added. Now save this file in pdb format. Save this file in pdb format. Here is the protein data bank file. In this format, right here, protein. This is what protein. So you have to save this file where? In docking folder which is created in C drive. So this is the protein. I will save it again. Yes, I will replace, so that protein gets saved in a docking folder. This one. I have taken only first this file 29x, that is the protein structure from protein data bank. Then I required configuration file for to specify the dimension along the XYZ axis. Then I have uh, prepared protein from this protein structure obtained from PDB-ID. Now the next part is you have to prepare the ligand. So how we can prepare ligand? We can prepare ligand by using ChemDraw. So I will open ChemDraw software. Uh, any synthesized compound that you have, uh, any compound that you have synthesized can be take as a ligand. Now I will draw simple chalcone structure. So this is what chalcone. So this is ChemDraw structure. I will select it. I will select it, copy it, and will paste in ChemDraw 3D. You will require ChemDraw 3D also for the optimization of ligand, because in ChemDraw, in ChemDraw, normal ChemDraw or 2D ChemDraw, we can draw the structure anyway. Suppose I want this ring here, I can put this ring here also. Just I will erase it. I will put just a minute. Okay, this is again chalcone. So the way by which you can draw the structure will not be correct always. So in order to get correct structure or the optimized structure, you have to paste that structure in this ChemDraw 3D, and then there is a tab for the energy minimization. You have to click on it. Now this lowest energy structure, we call it the optimized structure, is formed. This is what optimized structure of our synthesized compound. Now you have to save this file. There are number of things you have to do in molecular docking. You will not get in one day. You have to practice it for many times. Then and then only you can perform molecular docking. But once you learn how to dock, suppose I have created one folder TP. Just a minute. You have to select. You have to save it in that folder. But already I have created. That's why I am placing it anywhere on the desktop. So suppose there is folder. I will select it. Now this file will get saved in that folder TP. Save it with the name ligand. For this, I have kept this L in capital letter. L ligand. This is again important thing. Why? I will tell it later. So I have saved this file. I have saved this file in SD file format, in SD or SDF file format as a ligand, as a ligand. But remember, you will require ligand and protein files in PDB format. From ChemDraw 3D, you will get file only in SDF format. So you have to convert this SDF file format into PDB format for the docking in AutoWinner dock. I told you there are two important steps. First one is a protein preparation, and second one is ligand preparation. For the protein preparation, we have taken protein from the protein data bank. We prepared it in BioWare Discovery Studio and saved it in PDB format in docking folder in C drive. Then after we have draw the structure of desired compound in ChemDraw, then optimize it in ChemDraw 3D, and then we have to save this file format as a SDF file format in SDF file format, and at last, as we require both file in PDB, so at last you have to convert that SDF file format into PDB file format. For that, there is one more software, we call it as OpenBabel. There is one more file format, we call it as OpenBabel. You have to download it again. All these softwares are freely available on internet. You can download it. So I have downloaded already OpenBabel. So I will open it. OpenBabel. This is OpenBabel. Every time it requires installation, I will install it. So it gets open. This is input interface. This one is output interface. So here are three dots. Just click on it. It will open the previously selected folder. So we selected, we saved that protein, sorry, ligand structure in SDF format. That will be here. So this SDF format can be converted into PDB format by using OpenBabel. I will open this as an input file. Then I have to save it in the same folder, but this time in which format? Protein data bank format. Remember, this OpenBabel can convert more than 100 file format. 100 plus file format can be converted or interconvert from one file format to other. This time we required SDF to PDB, so I have selected here PDB, and then I will name it as again ligand, but this time for the simplicity I kept L in small letter, ligand, and I will save it. Okay, and then I click on convert. So this molecule get converted, that is in from PDB format get converted into, not from SDF format get converted into PDB format, and that will get saved in the same folder. I have copied these two and pasted in this, that means docking folder. First of all, we are having protein structure only, then we added here configuration file, then after uh, from this protein structure we prepared the protein, then by using ChemDraw software we have prepared our ligand in two formats. First one was SDF and second one was ligand. So our ligand can get open in this BioWare Discovery Studio. Can you see it? Can you see it? This is
Yes,
our
sir.
chalcone.
Yes, yes, sir.
Okay, this is our chalcone. So it gets converted into SDF format. This is not 2D structure; it is 3D structure that required for the molecular docking. Now both steps are over. That is ligand preparation. No, we don't want to save it. Ligand preparation, protein preparation. This log file is not required. This out file is not required. First of all, we have this crystal structure. From this, we have prepared protein. We require this configuration file. The coordinates, XYZ coordinates, we have taken during the protein preparation, and at last we have prepared ligand. In from the first, we saved it as a SDF file and then converted into PDB file. Clear? Now we will go for molecular docking with Autovina Dock. Autovina Dock is again freely available software on internet. I have downloaded and installed. This is Autovina Dock. It will ask permission for the registration. So just click on remind me later. It gets opened. This is the interface of Autovina Dock software. So first of all, we have to take our protein in pdb format. Where is that folder? C drive docking. Here is the protein. Here is protein in PDB format. Ligand is again PDB format. This structure was taken from the protein data bank, was also in PDB format, and this is ligand in SDB. So we required first protein. We'll take it in AutoWinner doc. So this is complex protein structure. There are number of amino acids here. Okay. Now the different steps in here in AutoWinner doc. The docking, for the actual docking, you will require protein structure and ligand structure in PDBQT format. So far, I was saying you will require protein and ligand in PDB format. Now I am talking you will require both in PDBQT format. This particular AutoWinner doc software will prepare protein and ligand files in PDBQT format, and by using command line software, which is system software of Windows, that can interact both this, or that AutoWinner doc can uh, perform molecular docking with the help of that command line software. So first, I have opened this protein structure in AutoWinner doc. Then go to edit. Then here is the tab charges. You have to add Coleman charges. This much charges get added. Then again click on edit, then charges, and again click on compute gastager charges. These gastager charges according to protein get added. Again I will click on edit, then atoms, then assign AD4 type. So listen, these are the number of steps. You have to just remember what you have to do. I think right now there is no need to know why we are doing this. Just understand what we are doing. In AutoWinner doc, we have added charges, Coleman charges, Gastager charges, and in atom tab we assign AD4 types. Then after, you have to save this file as a PDBQT format. So click on file, then save. The fourth option is write PDBQT. So click on atom, then press shift button, then uh, click on end. That means from atom to end all get selected, and then add. Okay. What happened? We have opened this protein structure in AutoWinner doc as a PDB file. This AutoWinner doc converted that PDB file into PDBQT file, and that gets saved in the folder. You can select the path also, but I have not selected. That's why that PDBQT file gets saved in this folder in C drive again, in the folder Windows 10. This is the protein in PDBQT. See, this is here is the timing 1 past 58. Here is again 1 past 58 something. So recently this file get created in this folder. I will cut it from this folder and will paste again in the docking folder. Got it? What I have done, I have cut this protein file, or we can you can simply copy paste it from that folder to this docking folder. So the most important step, protein in PDBQT got here. Now you got protein in PDBQT file format, required ligand also. So click on ligand, input, open. Now again C drive, this PC, C drive, there is docking folder, and now we have to take ligand, but the ligand available in PDB format. So you have to change your file format from PDBQT to PDB and select this ligand. I have kept here uh, ligand word with a small l, small letter. Okay. Now the ligand get opened in this window. Where is ligand? I have to search it. Here is ligand. Here is ligand. Can you see it? Can you see it?
Yes,
Hello.
sir. Yes, sir.
Okay. See, ligand is far away from the protein. Ligand is far away from the protein. So the what will be the next step? You have to insert this ligand into the active site of protein. Which active site? The dimension of active sites we have kept in configuration file. Okay. So there is again uh, steps that involves in the preparation of ligand. So click on ligand, go under the tab torsion tree and click on detect route. Then again ligand, torsion tree, choose route, and then again ligand output save as a PDBQT. So again the same folder docking. I will put here dot PDBQT dot PDBQT. So it gets saved in the same folder. Now the last step, which is important, that is nothing but molecular docking. For that, you have to select protein and ligand. So for that, go on the tab grid. Under grid, there is macromolecule. So far, we are choosing open option. Now, under macromolecule, we have to take the option choose. There is like a small window will open. We will click on protein. First, we will select protein. Select. Okay. Then there is generation of new file. It will require location. So we will place it in the same folder, that is docking folder. I will click on save. He is asking protein PDBQT is already exists. Do you want to replace it? Yes. See, the color of protein gets changes here. It was in red color. Now it is in bluish green color. Okay. Now second step, grid, set map types. Again, choose ligand and you have to click on ligand. First of all, firstly, we have selected protein. Now we are selecting ligand. Click on ligand. Okay. Now ligand also gets selected. The part of autotoxina is over. Now we will go for docking, and for that, in our computer, there is there is a software, command line software. We call it as command prompt. Just type here command prompt. You will get this here. We will open it. We will open it. Huh. Before that, just click here. You have to copy this file location or the folder location. So just click on here, right click and copy. After copying this, I will reopen that command prompt window. Okay. And I will type here cd space control V and enter. What I have done, I have copied this file location from this folder or a folder location and then open command from interface. Then I type here cd, then space, then I pasted this location of this folder and hit enter. Then I have to give the command here vina.exe space dash dash config space config dot txt space dash dash log space log dot txt and will hit enter. Now the docking process started. It is reached up to 100%. Okay. Now the docking is done. What you see here? What you can see here? Yes, yes. What you can see here. Hello, am I audible?
Yes,
Am
sir.
I audible? Okay. You
Yes,
will
sir.
get your
You are
output.
audible.
You will get output file here. You will get two new files. First one is a log file here, and second one is out file. Now this out file includes, where is command prompt? This out file includes the 9 different poses of ligand, but they are in one file. We have to separate it. So there is one more command is remaining. Just type here vina underscore split dot exe space, then input, sorry, dash input. See the changes what happen in this folder. There is out file, input space out dot pdbqt, and we'll hit enter. So see here, there was only one file as a out file. Now you will get out one, out two, out ligand one, two, three, up to out ligand nine. That means you got nine different poses where ligand can interact with what, our protein. This is output file or log file where you will get this docking score. It was a minus 8.8 kilocalories per mole. We call it the binding affinity with the RMSD value 00. So we can report it as a docking score, and you got the different docking poses. Now, so far the docking is done. Now you have to analyze the docking results. How? By using BioVia Discovery Studio. And for that, just click on a protein. Just click on protein. Again, the protein. This protein structure you have prepared already. Now I will open the different types of ligands here. Click on file, then open. Here are different options. So I will select here all files. All files, you will get this out ligand 1, open. Now the ligands get open in this structure. Here ligand gets open in this structure. Okay, I will open another one. That I have reported in, I will open it in this particular. Wait, we can directly open file, open, type document. Okay, all files, out ligand 1. Okay. So I have taken this ligand that I have reported or presented in what we can say PPT. So you will get maximum interactions. I will copy this ligand and I will paste here. So what happens? Our ligand get inserted into the active site of this protein. Can you see here? This one. This is our ligand that we have added. Can you see it?
Yes, sir.
Can you see
It
it?
is clear. Yes.
Okay. Okay. Uh, for the clear visualization, we will go for publication quality visualization. See, whenever you will require the images, go in the tab script, there is visualization, and there is one option publication quality. You will get this results in publication quality. So you can clearly see this was the heme group, and this is our synthesized compound. This synthesized compound get inserted into the cavity of this protein. Now we can see its interactions. How? Just click on this, go here, view interactions. You have to select this ligand as a, okay. We will select this ligand. This ligand gets selected and go for ligand interactions. See, there are number of interactions of the ligand that takes place inside this cavity. Inside this cavity. I have already prepared that. We will go for that also. Vanillin 14. Yes. Or we can also open another one. Docking. Okay. Okay. This is without him. No, it will not be good for the docking. Okay. Yes. Close. No. No. Just, no. At a time, two windows get open. This is protein. Again, we will open ligand. 3A. All files out. Okay. This is our ligand. We'll copy it. We'll paste in hierarchy of protein. Paste. Go for script visualization, and there is publication quality. So this is our ligand. Ligand 1. We will select here ligand 1. And now go for ligand interactions. Yes. These are the number of ligand interactions with the protein. Just click here receptor. You will get this type of images. You can see here this ligand have interactions with heme unit also. Here is the heme unit, and here is our ligand. So again, go for script visualization and publication quality. Visualization and publication quality. Okay. No, actually ligand was selected. Again, script visualization publication quality. Okay. Now you can label the different types of amino acid residue. How? Just remove this receptor. it get removed. These are the amino acid residues. I will click on one by one and go for label, add. We can know the which type of amino acid residue is there. I will select here amino acid residue or amino acid only. Here desired font size. I will take here 24. I will give whatever color I want to represent. Apply. Okay. Now it is lysine 74. This residue was lysine 74. I will click here, then right click, label, add. I will add the name of residue here, methionine. Again, here, label, add, apply. Okay. History knows there. Again, I will click here, right click, label, add, apply. Okay. Which one is remaining? I think this one is remaining. Click here, right click, label, add, apply. Okay. I think all residues are labeled. This one is remaining. Label, add, residue, apply, arrange. Okay. Label, remove. Again, label, add. Here is residue. Oh, and this is the heme unit. This one was heme unit. So these are what number of amino acids that directly interacted with our ligand or synthesized compound. All these are non-covalent interactions. So I can generate. Now we can add the receptor protein whole that was just interacting residues. These are only interacting residues, and this is whole protein. So I can see its 2D structure also. Before that, I will also format this ligand as a ball and stick. Here I can put here whatever I needed. Again, color can be changed for the ligand. If I want this one is good, I will take apply. Okay, okay. Now in this way we can visualize our ligand protein interactions. Whatever you want, you can change. If I want, I can change this also. This style. If I thought it should be like this. Now, now I can go for 2D diagram that you require for the publication. This diagram I was reported in PPT, that's why I have taken this one. So again here you can change the display format. Here suppose I want a ball and stick model. I will keep it as the stick size same and so ball size. Then here I want this, apply. Okay, this is our 2D diagram. This is our 2D diagram. So this interaction can be analyzed and can be report for the publication. Again, if I want, I can see the number of informations like molecules, cell, amino acid chain, amino acid, atoms chain, unfavorable non-bonded interactions, unsatisfied non-bond label. This is the information we can collect it and we can report it in a result and discussion section of our research paper. Again, we can change the color of this whole protein also. Whatever we want, we can do it. Huh. And at last, suppose uh, this image is or this position is good. Now go for file, save as, just click on here image file and whatever you want. Suppose I will take it on desktop, TP 3D image. I will save it. Whatever dimension you can take here and okay. This is the 3D. This type of image you will get. This is the publication quality image. You can report it in your research paper. Again, the 2D image you can report here, showing like this. Just go in file menu, save as image file. It is 2D. Save it as 2D. Cancel. No. File. Save as in the desktop. TP. Save. Okay. It gets saved in this folder, separate folder. So this is our 2D diagram. So these images can be very useful for the your research paper, and the related information you can report in result and discussion section. Okay, if anyone having any queries or the doubts, I will be very happy to answer that. I think the most of part related to docking is covered. Yes, from the participants, anyone having any doubt? Hello, hello.
हा सर बोला.
हा, कोणाला काही डाऊट असेल तर सांगा, नाहीतर झालाय पार्ट डॉकिंगचा.
Thank you. If anyone have question regarding the session, please drop down into the chat box. Anybody have a question? Sir, I think no question in chat box.
Okay, sir. Okay. Then over to you, sir, for the.
Thank you, sir, so much for wonderful and insightful session. In this session, sir, discuss three things. What is molecular docking? Why study of molecular docking is important, and how we perform the molecular docking. Sir also explain the types of docking programs, visualization tools, workflow of molecular docking, and how the protein and ligand preparation takes place. Sir also shown visualization of molecular docking, and from that we clear how the protein and synthesized compound interaction takes place. Thank you. Now,
Thank you so much.
now it's time for vote of thanks. On behalf of organizing committee, I express my sincere gratitude to Dr. Rahul Shinde sir for his valuable guidance on molecular docking. I also would like to thank Honorable Principal Dr. Jagdale sir. And last but not the least, thank you all the participants for joining us today. And from with the grand permission of organizing committee, I conclude the session. Thank you. Thank you all.
Thank you, sir.
Okay, thank you.
Can I leave?
Yes,